like a snail shell actually has a portion of the temporal bone that sort of supports it. Okay? So it's not just all made up of soft tissue. There's actually a bony support for the cochlea. Okay? So someone asked me the other day, and it's something that I always struggle with. What does this mean, this bony labyrinth? And it just means that there's a space inside the temporal bone where this exists. What is meant by membranous labyrinth? This is the cochlea, or a portion of the cochlea. The cochlea is made up of membranes, basically, and they're fluid-filled. And so this is a space inside the cochlea that starts here and makes its way all the way through. So inside the cochlea, there are actually membranous spaces, and those things are called the membranous labyrinth. Okay? All right, so what do we have here? We have, in these things here called the semicircular canals, we have a membranous labyrinth, and it is filled with a high potassium fluid known as the endolymph. High potassium is important because in the receptors for hearing and balance, the depolarizing ion is going to be potassium, not sodium. Okay? So high potassium fluid here. This inner portion of the cochlea is also called endolymph, this fluid in there, and it's a high potassium fluid. What you're going to find by the time this is over is that the receptors for sound and the receptors for balance and equilibrium are built on a similar architecture. Okay? They're both going to use potassium as their depolarizing ion. They're both going to use mechanical gates to open ion gates. Okay? All right. Any questions about what you see up here? We will look at this thing a little bit more closely. All right? Okay. Looking at it a little closer, we saw, a few of us saw this in the lab. There is the cochlea, and if you were to do a cross-section of the cochlea, you would see then the three places where fluid exists. Okay? This is called the vestibule, and there's something going on in there that we'll look at later. Those are the semicircular canals as you've seen. This fluid-filled container here is in contact with this space right here called the oval window. Anything that pushes on this oval window is going to create pressure waves and some fluid movement in this membranous space. There's a fluid in here, and if you push on this oval window, it is going to lead to fluid movement and pressure waves in here. This cylinder of fluid makes its way all the way around the cochlea until it finally reaches the tip. Okay? Until it finally reaches the tip. When it reaches the tip, which is called helicotrema, or trema, when it reaches the tip, whatever movements of fluid or pressure that did not directly affect this spiral organ, whatever did not affect the spiral organ will be lost then to this area, this particular fluid-filled membrane, or membranous location. Because at the helicotrema, this fluid-filled cylinder and this fluid-filled cylinder are in contact with one another. They are separate throughout the whole cochlea until we reach that point there. Why do you care about such a thing? Because the fluid movement in here is going to have an effect on this device. And the fluid movement in here is going to be related to the frequency of sound that is occurring at the time. If the frequency of sound is either too high or too low to affect this device, then that fluid movement, that pressure that is generated by that frequency of sound is lost. Low frequency noise that does not affect this device is lost at the helicotrema, and it will lead to then some vibrations at the round window, and the low frequency sound will just get lost. okay? High frequency noise goes under a different scenario. But either way, whatever's happening in here eventually makes it over to here. This thing is sort of like a fluid-filled balloon. So if you were to have a fluid-filled balloon and squeeze it on the one side, the other side would grow. You've done this, correct? That's what the relationship between these two parts of the cochlea. As we push here, we get some reverb or vibration here. The movement of fluid is dampened off at this location. All right, so this thing here is called the spiral organ, and it is a separate fluid-filled container. It is called the cochlear duct. That's where the fluid is located, and you see that it's called endolymph inside there, high potassium fluid. The receptors for sound are right here. They are cells which generate what's called receptor potential based upon how much potassium comes in to the cell, okay? They are synapsed, these cells, with neurons. The neurons will congregate in a cochlear ganglion. There'll be some sound processing right there, and then the cochlear ganglion will have axons that leave it, and they will become the acoustic nerve, otherwise known as the vestibulocochlear nerve. Okay. So you'll see in lab today that the eardrum, the tympanic membrane, as you hear my booming voice, it causes vibrations in the tympanic membrane. That is the eardrum, which is the beginning of the middle ear. The tympanic membrane is jointed with the first of the 3 ear bones. The generic term for these ear bones is called the ossicles. The first of 3 is attached to the tympanic membrane. As the tympanic membrane moves, that bone moves too. The movement of the first ear bone then makes its way through the second, through the third, and the movement is actually amplified. So what could be a little bit of movement in the tympanic membrane actually becomes a lot of movement in the third of the ossicles. Okay? The third ossicle is stuck in the oval window. So when the third ossicle vibrates, it pushes on the oval window. Okay? And that sets cochlear fluid into wave motion. Pressure waves move through the fluid known as the perilymph, which fills those blue cylinders. We'll go back to it in a second. Okay? It moves through that fluid. Let's go back. This is connected to the oval window. It's called the scala vestibuli. The oval window lies in the vestibule of this inner ear apparatus. It contains a fluid that is similar to CSF, perilymph. When the oval window receives vibrational movements, there are pressure waves in here. Those pressure waves pass right through this little membrane here, which is called the vestibular membrane. This thing is so thin that it really provides no barrier to the pressure waves transmitting their strength from the scala vestibuli right into the cochlear duct. This membrane here, called the basilar or basilar membrane, is going to be constructed of a connective tissue. The connective tissue is going to create a platform on which the sensors of the ear rest. The basilar membrane will deform as pressure waves come across. Which portion of the cochlea deforms depends on how the basilar membrane is constructed. And this is one of the keys to hearing sounds at different frequencies. In some portions of the cochlea, the basilar membrane is strung real taut, like guitar strings, okay? In other portions of the cochlea, the basilar membrane is strung a little bit more loosely. So the pressure waves are going to move across where they can. If the basilar membrane is strung more loosely, then certain frequencies of sound are going to deform it versus others. Why does this matter? By deforming the basilar membrane, we are going to affect these sound receptors here. We are going to open up ion gates, and we're going to depolarize them. So in a portion of the spiral organ that has a basilar membrane that is tuned to a particular frequency, when that frequency comes into the ear, it's that basilar membrane that will vibrate, and it is that portion of the cochlea that will send action potentials to the brain. The brain will know that it was that portion of the spiral organ which is tuned to a particular frequency, and the brain will know then what that frequency is. Now, what is frequency? If you are like me, which none of you are because you're not as old as I am, you remember the days of your stereo being the biggest and the loudest on the block, okay? And so we got into the sort of the technical aspects of audio, okay? We tried to understand, what we were trying to reproduce in our speakers. So you learn about frequencies that humans can hear. And basically, humans can hear frequencies that vary from 20, and the unit is hertz, Hz, to, I think there is some disagreement as to how high we go. From 20 to 22,000 hertz. Any sound below 20 hertz we don't hear. Any sound above 22,000 hertz we do not hear. Are there other animals that can hear beyond this? Yes. Okay? A dog whistle is based on high frequency noise that humans can't hear, but dogs can. This is low frequency sound. Boom, boom, boom. The kind of sound that passes through walls. Okay? This is high frequency sound. The tweeters. Actually, I shouldn't say tweet anymore because it means something different. Tweeter is the type of speaker that produces high frequency sound. Okay? This is really the kind of sound that is really, really high frequency, really, really high pitched, I guess, does not pass through walls very well. So it has sort of a lighter energy associated with it. So your cochlea has a spiral organ that is tuned to these frequencies. All right? Okay, let's take a look at that. There it is. So here, the author of this diagram has taken a cochlea and unraveled it. Okay? And there is the scala vestibuli, and there is the third ossicle. What's that ossicle called? Stapes, okay, or stapes if you want to. And you can see that there's a little bit of vibration going on here. You can see the tympanic membrane. This is called the malleus. This is the first ossicle. This is called the incus. This is the second ossicle. And that is the stapes. By the way, this is a synovial joint. It's meant to be able to move all the time. So there's the tympanic membrane, and then there are the three ossicles, and then there's the oval window. So in this particular diagram, what we are seeing is high frequency noise, okay? And in this case, we're seeing a lot of noise coming in, but here we are seeing the pressure waves moving through the scala vestibuli and deforming the basilar membrane. So this area of the cochlea then is tuned to that particular frequency. What you're seeing here then is low frequency sound being detected by the cochlea farther on down the line. So the basilar membrane here is constructed a little bit differently. It's a little bit different. It responds differently. And then here they're showing any noise that we cannot detect. It makes its way around the helicotrema, comes around the scala tympani, and then dampens off at the round window. Okay? So this is something that you're going to want to study because you have a test question that asks for this particular information. Do you get this then? in summing it up, it all comes down to